Hello everyone, I'm Praveena from Group 320. I'm going to explain about the physics of proteins, the protein, globules and hydrophobic interactions and relation between the primary and the three-dimensional structure of proteins. First, the protein globule and hydrophobic interactions a protein globule forms when a protein folds into a compact 3D shape. It has the structure and flexible parts due to the different types of amino acids like proline, which cannot form strong bonds. This folded shape is essential for the protein by the function properly in the bonds. Like if we talk about the examples like lactase, which breaks down lactose, the shape in and proteins can have a quaternary structures, meaning they are made of multiple smaller globules. For example, hemoglobin has four globules. The proteins coat of the tobacco mosaic virus has about 2000. These globules comes together automatically through a process called self-assemble. Proteins folds in a compact shapes mainly due to the hydrophobic effect, where water pushes away non-water loving parts of the protein. It helps to create a stable structure for the protein to function in watery environments, while strong connection called disulfide bridges provide some stability. Weak forces were hydrogen bonds and the hydrophobic effects are crucial for shaping the protein globule. If we talk about this equation, it shows that the chemical potential is calculated based on the energy, state, and concentration of the substance in each environment. It highlights the relation between the substance energy concentration and how it behaves in different environments. And next equation, if we talk about this one, indicates that the chemical potential of the hydrocarbon in CCl4 is lower than the water. The negative value suggests that the hydrocarbons prefers to be in the non-polar environment CCl4 than in the polar environment in water. Next equation, if we talk about this one, the positive charge that the dissolution of butane in water is not favored thermodynamically, meaning unfavorable in condition. It is an unfavorable in condition, and overall decrease of the entropy and release of heat both contribute to the changes in free energy when butane dissolves in water. Like if we talk about some are important to note that the hydrophobic residues can also be found on the surface of the globules. This presence can influence proteins interactions with the surrounding environment, and some hydrophobic regions may still be exposed to water, affecting the proteins behavior and functionally. Next topic is the relationship between the primary and the 3D structure of proteins. The building of biological molecules and supermolecular structures invariably involves two stages like biosynthesis of large and small molecules. Proteins are and these proteins are made of long chains and amino acids. Some amino acids are hydrophobic, water repelling, and stick together, while others are hydrophilic, water attracting, and stay separate. Examples, if we talk about this, insulin protein regulates blood sugar levels and it's crucial for its function called special sequence of amino acids. And next example is collagen. This protects and provides strength and structure in skin and connective tissues. This example shows how the sequence and arrangement of amino acids in proteins determine shape and function. And the formation of alpha helix requires energy at the beginning, but once they start to grow, no extra energy is needed. The formation of alpha helix requires energy at the beginning, but once they start to grow, no energy is needed. The organized approach to folding simplifies protecting the protein's final structure, particularly for proteins that are composed of either alpha helix or beta sheets. In proteins, folding longer structures like alpha helix and beta hairpins are more favorable than several short ones. Longer ones like to form a stable double helix structures, and longer beta hairpins are preferred over shorter ones. The theory has been validated by the experiments such as those conducted on ribonucleus and suppose the idea that denaturation helps in a cooperative manner. The number of amino acid changes per residue over a set time varies significantly among proteins, showing that the proteins can evolve primarily through neutral mutations and random genetic drift. Natural selection tends to act more on the overall organization traits phenotype rather than on the proteins themselves. However, this theory has faced criticism for conflicting with other environment concepts. For example, like that the biological functions of proteins, like that the biological functions of proteins is determined by its three-dimensional structures. The extension of unanimous of the correspondent is not clear at all. It is believed that the changes in the primary structure of a member, like biological functions of these proteins, in other words, mutations, and then the neutral or natural selections. This forms the basis of the neutralistic theory of evolution, advanced by the Kimura in 1968 by analyzing the evolution of homologous proteins. Kimura came to the conclusion that first, for a particular protein, the rate of evolution expressed by the number of amino acids replacements per geological unit of time, it is nearly constant and is same for different evolution parts, being subject to the relatively smaller functions, and constants undergo evolutions faster than molecules characterized by strong per amino acids. Residues to the 10 years are as follows: fibrin optates of chains of hemoglobin, cytochrome, histone, H4. Thus, molecule evolution predominantly involves like neutral mutant mutations, the random drift of genes, natural selection. Natural selections and the random drift of genes operates at the phenotype level rather than at the level of bipolymer molecules. The neutralistic theory met with a sharp objects as contradicting. In modern Darwin theory of evolution, as a matter of fact, the contradiction exists between the neutralism and the selectivism. The physical essence of neutralistic theory consists of a primary and the biological functions, like three-dimensional structure of the proteins. This is the ending of my topic. Thank you.